We will be discussing on cDNA library, that is a part of the course curriculum of UG Botany, MSc Biotechnology and MSc Life Sciences. This is a pictorial representation of the difference between the preparation of a genomic library and a cDNA library. For the genomic library preparation, the whole DNA is extracted. In a cDNA library, the mRNA is extracted and cDNA is synthesized from this mRNA. After this, the steps are basically same. So the DNA is subjected to restriction digestion. Then the pieces are ligated to a digester vector. The digester vector is amplified in a host or by PCR, and in case of a cDNA library, the cDNA is ligated to a vector directly, so this restriction digestion step is not required in the preparation of the cDNA library. We will be discussing the topic under these headings: introduction, genomic and cDNA library, the basic difference, genome and transcriptome, what is that? Extraction and purification of the mRNA, because that is the starting point of cDNA library. Synthesis of double-stranded cDNA, addition of adapters to the cDNA, vector digested by RE, how it is digested and why it is digested. Then ligation of the double-stranded cDNA to the digested vector to create the rDNA, recombinant DNA. Then how the rDNA is used for transformation of the host, then selection of the transformants and the storage of the colony. A cDNA library contains genetically engineered bacteria and bacteriophages, and these bacteria and the bacteriophages which constitute the cDNA library contain engineered vectors. The engineered vectors have cDNA, that is complementary DNA. In a cell, mRNA is the active transcript that contains only the coding sequence, and the DNA that is synthesized from this mRNA by reverse transcription is called as complementary DNA. So we can tell that the cDNA library contains only the coding sequences of an organism or a cell or a tissue. Now the basic difference between a genomic library and a cDNA library. The genomic DNA library contains both coding as well as non-coding sequences. But in a cDNA library, only the sequences which are represented by the exon in case of eukaryotes, or the sequences available in a transcript are there. So that is the difference between the cDNA library and a genomic library. This picture shows the flow of genetic information and some basic concepts of molecular biology. Genome represents the total amount of DNA of an organism or haploid DNA of a cell. Now transcriptome. Transcriptome is the total amount of mRNA of an organism or a cell. Proteome, the total amount of proteins of an organism or a cell. Genome represents the coding genes, non-coding genes and the organelle genes. Transcriptome represents the protein coding mRNA and tRNA, rRNA, which are non-coding RNAs. Then proteome, it represents all the types of proteins including the enzymes. Metabolome is represented by amino acids, sugars, lipids, all the metabolic products and nucleotides. Now, genome. Genome actually represents the total number of annotated genes. Annotated genes means the genes with a designated location. So that makes the genome. And transcriptome is the total amount of active mRNA in a cell, translatum. Out of the total amount of active mRNA, the highly active mRNA which are functional at a particular moment are called as translatum, and proteome and secretum includes the mRNA, those are engaged in protein synthesis, that is the proteome. This is another way of representation of these terms. Now, what are the requirements for the construction of a cDNA library? The primary requirement is pure mRNA that is not contaminated with any other molecules of a cell. Then the enzyme reverse transcriptase that will carry out reverse transcription. Once we get cDNA, the complementary DNA, it has to be modified as per requirement by the ligation to a adapter, linker, or by homopolymer tailing. Then the vector. Vector may be a plasmid or a phasemid or some phase-derived vector depending upon the host in which the library will be propagated. The vector is digested with the restriction enzyme. cDNA is ligated to the digested vector to get recombinant DNA, and the recombinant DNA is propagated in chosen vector, either a bacteria or a bacteriophage. Then it is stored under certain conditions, under the required conditions for further use. For cDNA synthesis, the primary requirement is pure mRNA. mRNA in eukaryotes and prokaryotes have some differences. In case of eukaryotes, the mRNA consists of a poly A tail at the 3' end, but in case of prokaryote, mostly, not always, mostly in case of prokaryotes, the 3' end does not contain a poly A tail. Even if there is a poly A tail, that is very short and that is unstable. So for the purification step of obtaining RNA, mRNA, there is a difference in procedure for prokaryotic mRNA and eukaryotic mRNA. Bacterial mRNA usually has a very unstable poly A tail or a very short poly A tail, or there is no poly A tail. So poly A tail cannot be used for the separation and purification of the bacterial mRNA. There is a technique, that is magnetic capture hybridization method, that can be used for the purification of bacterial mRNA. For this technique, two things are required. One is the capture oligonucleotide, means the oligonucleotide which are meant to capture certain target molecules. So the capture oligonucleotides are mixed with the purified RNA mixture and it is poured into a tube. The magnetic bits which are chemically modified or derivatized with oligonucleotides are next added to the tube. The magnetic bits have certain oligonucleotides which has affinity for the capture oligonucleotides. So when all these are mixed together, after some time what happens? The magnetic bits will capture 16S rRNA and 23S rRNA as per the kit, or different forms of rRNA or tRNA, because they have the target molecule, that is 16S rRNA, 23S rRNA or other types of RNA, but not mRNA. That what happens? mRNA cannot bind to these magnetic bits, so mRNA remains in the supernatant and it can be eluted from the supernatant. cDNA library is actually a collection of bacteria or bacteriophages, and each host has a piece of cDNA or complementary DNA. So this is the overview of the library construction. A source is selected, a cell or tissue or from some organ, cells are selected, and from these cells the RNA mixture is extracted, and from that mRNA is purified. This is the purified mRNA. Purified mRNA is used for the synthesis of double-stranded cDNA by using RT-PCR. Then this is the blue one is the digested vector that is ligated to the cDNA. Each is a different piece of cDNA. So in the next step, the recombinant DNA is introduced into the host cell. Host cell is multiplied in a culture plate, and now this plate contains colony of this bacteria, and in each cell of the bacterium there is a piece of this particular piece of cDNA. So these plates together, if we combine all the cDNA and all the plates, that is called as a library. This is the detailed view of the construction of the cDNA library. So here the first row, it shows different mRNA molecules and they are designated by different colors. These are the active transcripts at a particular time. They are extracted from the cell. The mRNA is used for the synthesis of different pieces of cDNA. So these are the mRNA, then these are the cDNA synthesized from each mRNA. These are double-stranded cDNA. The double-stranded cDNA are ligated to the vector. Vector is introduced into the bacterium, then the transformed bacteria are selected and propagated in the form of plates. So each cDNA is propagated in a separate plate. So if we combine all the plates, it represents a cDNA library. Here is the protocol for the isolation of mRNA of the eukaryotes. The eukaryotic mRNA have a poly A tail at their 3' end, and this property is used for the separation and purification of the mRNA of the eukaryotes, and it is done by affinity column chromatography. The column is made up of cellulose that is covalently attached with the oligodeoxythymidine chains. Oligo-DT has affinity for the poly A, so they form complementary base pairing. So when a mixture of different RNAs is poured through the column, mRNA sticks to the column bits and other RNA get eluted because they cannot bind to the oligo-DT. After some time, again 0.5 m NaCl is used as the washing buffer to elute the unbound RNA molecules, and finally mRNA can be collected by using a solution of low ionic strength or water. This is a close-up view of the separation procedure of the eukaryotic mRNA. So this is the mixture of mRNA molecules, and the mRNA molecule mixture is contained in the tube, then poured through the column, and these are the bits, oligo-DT, cellulose bits, and the red ones are the mRNA molecules. They stick to the bits because of complementary base pairing between poly A tail and the oligo-DT, and blue ones and the other black ones are the different other molecules of RNA which are eluted, and finally the bound mRNA can be extracted by using a solution of low ionic strength. Magnetic beads can also be used to get pure mRNA. So here what happens, the beads of the column are made up of some paramagnetic materials, cellulose beads, but they contain some magnetic property. The oligo-DT column that is filled with the magnetic beads, it is attached with the protein streptavidin. Streptavidin is a protein isolated from the bacterium Streptomyces avidini. Then mRNA is attached with biotin or vitamin B7. So why it is used? Because biotin has affinity for streptavidin. The column beads, the magnetic beads contain streptavidin, and mRNA has biotin. So naturally what happens when the mRNA passes through the column, the mRNA with biotin, that is biotin-ylated mRNA, sticks to the streptavidin of the magnetic beads. And the affinity between the biotin and streptavidin is so high that mRNA will bind to these beads with very high efficiency. At the end, a magnet is used to collect the beads to one side. Then the beads are washed to get the mRNA. So this is how the procedure works, the magnetic capture technique. The beads are made up of some magnetic materials and they are conjugated with the protein streptavidin. mRNA is conjugated with the biotin, that is vitamin B7. Streptavidin and biotin have affinities. So this is the picture of a magnetic bead and this is the oligo-DT bead. So this is the oligothymidine. This is the poly A tail of the mRNA. So mRNA with the poly A tail binds to the beads, but these beads are different from the normal beads because they are made up of some paramagnetic material. There is affinity between the streptavidin and the biotin. Therefore, the binding of the mRNA to the beads is very strong. The other RNAs are in the supernatant. They do not bind to the beads. So in the next process what happens, the other RNAs, rRNA, tRNA, etc., they float in the supernatant, and when a magnet is used from the outside, the magnetic beads along with the mRNA are drawn to one side of the tube. Then finally they can be recovered from the tube. Once pure mRNA is obtained, the next step is to prepare the first strand of cDNA, followed by the synthesis of the double-stranded cDNA. And the requirements are the mRNA template, the enzyme reverse transcriptase, and in some cases DNA polymerase, then dNTPs, buffers, S1 nuclease, and RNase H enzyme. There are different methods of the synthesis of the cDNA. So this is the mRNA molecule with the poly A tail, 5 prime, this is 3 prime, and oligo-DT primers are added. Then in presence of the enzyme reverse transcriptase and 4 nucleotide triphosphates, the deoxynucleotide triphosphate, the first strand is synthesized. So here this is the hybrid RNA DNA molecule, that is mRNA cDNA hybrid molecule. Now to digest the mRNA molecule, RNase H is used, and RNase H cuts the mRNA molecule from a cDNA RNA hybrid, and these small strands of the mRNA now serve as the template. And using this template in the presence of the 4 deoxy-NTPs, the second strand of the cDNA is synthesized. Now this is the picture of a double-stranded cDNA. There is another procedure for the separation of the cDNA from the RNA-DNA hybrid, and this involves homopolymer tailing, alkaline sucrose density gradient, then the enzyme terminal transferase, and finally cDNA can be separated. In the next slide, we will have a detailed view of this procedure. This is the mRNA molecule, the poly A tail in the 3' end, By using reverse transcriptase and the 4-deoxy-NTPs and oligo-T primer, the first strand of cDNA is synthesized. In the previous procedure, RNase H was used to degrade the mRNA from the first strand of cDNA and mRNA hybrid. But all the reverse transcriptase do not have intrinsic RNase H activity, and in that case homopolymer tailing can be used. So this is the picture of a RNA DNA hybrid. Now terminal transferase and dCTP are added to the 3' ends of the mRNA molecule and the first strand of the cDNA molecule. Then after this step, both the strands are subjected to alkaline sucrose density gradient centrifugation, and when this procedure is done, the mRNA is totally removed and a pure cDNA strand is recovered. This is the first cDNA strand. Then in presence of reverse transcriptase or DNA polymerase and the other requirements, the double-stranded cDNA is synthesized. This is another procedure for the synthesis of double-stranded cDNA from the first strand of the cDNA. So here this is the mRNA molecule. The mRNA molecule with poly-A tail, by the usual procedure, the first strand of cDNA is synthesized, but the reverse transcriptase, it extends beyond the 5' end of the RNA and creates a short loop of the cDNA. After this, the activity of the reverse transcriptase stops, and now this loop area becomes double-stranded, and once there is a double-stranded region, then the reverse transcriptase or DNA polymerase can get attached here and it will start synthesizing the second strand of the complementary DNA, and finally the loop can be removed by using S1 nuclease, single strand specific nuclease enzyme. Once the double-stranded cDNA is synthesized, the next step is the preparation of the recombinant DNA by ligating a digester vector with the double-stranded cDNA. There is a wide choice of vector that depends upon the final objective. Plasmid can be used, phasmid, bacteriophages, so different types of vectors can be used. The vector is digested with the restriction in endonuclease, and after that the digested vector is ligated to the double-stranded complementary DNA to get recombinant DNA, and recombinant DNA is introduced into the host cell to transform the host. Then there is selection of the transformants, and after that the transformed bacteria are stored in the form of a library. This is the yeast artificial chromosome. This is a high capacity vector, and the markers are used to isolate the transformants. This is also a high capacity vector, the bacteria artificial chromosome, and here also there are the marker genes which help in the selection of the transformants. This is a plasmid vector, and again you can see the selectable markers that helps in the selection of the bacteria which are transformed with the recombinant DNA that was prepared in previous step. After the synthesis of double-stranded complementary DNA, the next step is the ligation between the vector DNA and the double-stranded cDNA to create the recombinant DNA. This is the plasmid DNA that is used as vector for preparation of a cDNA library in bacteria. The plasmid DNA is digested with a particular restriction enzyme, and we can see there are sticky ends generated by this procedure. And in this image, the double-stranded DNA is with blunt ends. the ligation between the blunt end and the sticky ends is not very efficient. To increase the efficiency, the cDNA is attached with linkers at both ends, and after that it is digested with the same restriction enzyme that was used to digest the vector DNA to create sticky ends. And now the sticky ended cDNA and the digested vector DNA, also with the sticky ends, are ligated in presence of the enzyme DNA ligase. After the construction of the recombinant DNA, next is amplification of the recombinant DNA. For that, different methods of transformation can be adopted. One such method is shown here. The bacteria is transformed by using the electric shock. Besides this, there are several other methods that can be used. The transformants can be selected by different procedures, either by the use of selectable marker genes like antibiotic resistance genes or reporter genes. In this diagram, this is the selection procedure by using selectable marker genes, and there are blue and white colonies. The white colonies represent the transformed bacteria that contain the cDNA. Once the library is created, the next step is to store the library properly for further use. So there are different storage methods that can be adopted: cryopreservation, or in glycerol, or with DMSO also the library can be preserved. And these libraries which are preserved in such a manner are all random cDNA libraries. To use the library, the random library is selected, then the bacteria are plated out on a plate, a nutrient medium, and once the cells grow, individual colonies are picked up one by one. So one colony represents one type of bacteria with one type of cDNA. So if we pick one colony, that indicates a mass of similar type of cDNA, and these are the microarray plates. The colonies can be arrayed or replicated in the microtiter plates and then screened for further use.